So Bismillahirrahmanirrahim. In the previous video, we were discussing something related to the hair cells. Now, in this video, we will discuss the function of utricle and saccule in the maintenance of static equilibrium. I told you in the utricle and saccule, the organs of it or the receptors were the macula or the hair cells, which are the organs of static position. Now, the hair cells of the macula, they are oriented in different directions. Some are oriented like inert, horizontal, some are vertical, and these positions not only help them in detecting the direction of the motion, but only one type of cell is stimulated with one specific type of direction of motion. Because of this different stimulation of different macular cells, the body becomes aware of the brain becomes aware of the type of motion that is taking place. So as a result of which, because of different cells being activated with different positions, the vestibular nerve carries these signals to the brain. Now the vestibular system, the cerebellar system, the reticular motor system, all of them, they get activated. They activate the postural muscles to help maintain the equilibrium in relation to that position whose cells are activated. And even when there is a half of disequilibrium, the half of the disequilibrium, it will be very precisely detected by the macular hair cells. The macular hair cells will send signals to the vestibular nerve, and the brain will be apprised of any sort of disequilibrium through these cells. That is how we are able to maintain our position. Now, what happens when the body is thrust forward suddenly? For example, if these are like hair cells, and this is the statoconia, or the position, or the statoconia, that is made up of calcium carbonate, as I told you. And the hair cells are inserted into this statoconia. What happens due to the body being moving forward? What will happen that you are in a car and your body is moving forward. These statoconia cells, now they are going, the statoconia, which is having specific gravity greater than the surrounding fluid, it will push on the cells and it will push them downwards in the direction of the motion. When it is pushing them downwards, what will happen, that there will be a signal generated which will be carried by the vestibular nerve to the brain, and brain will be apprised of the position that our body is in a state of disequilibrium. The brain, through the cerebellar system, through the vestibular system, through the reticular system, the brain will activate our postural muscles. Our postural muscles will try to pull our body forward. When we will pull our body forward, when we will maintain our posture, what will happen? The statoconia, it will come to its normal position. And when the statoconia comes to its normal position, the signals that were going through the vestibular nerve, they will now stop going. As a result of which, it means that the body has corrected its disequilibrium position to the equilibrium position. So macula, they are going to operate to maintain equilibrium during linear acceleration, exactly in the same way as they do in the static equilibrium. So apart from static equilibrium, the macula are going to help in linear acceleration as well, but not with linear velocity. So you need to clearly understand three things. That one thing is, they are going to maintain static equilibrium. They are going to maintain linear acceleration. Linear acceleration is something in which the velocity does not change, and sometimes it becomes zero even. But in linear velocity, the macula will not be able to operate for the detection of linear velocity or change in velocity. For example, when what happens is, when you are running, you have seen runners, they bend a little bit forwards. Why do they do so? Because if they were running in vacuum, they would not have to bend forwards, and they will have to start like in a linear position and then move forward. But when they run in the earth where there is air, because of the presence of air resistance, which can be detected by the cells that are located in the body, the pressure sensitive cells, so we need to bend forwards to overcome that air resistance. And when we achieve certain sort of acceleration, then our body can relax back to its normal position. Also, when we are bending forwards, what happens is that the statoconia and all the things associated with the vestibular apparatus, they are also apprised, and they keep the body forward to prevent it from falling backwards when we are running by the means of air resistance. So this is how detection of linear acceleration is done by utricle, saccule, macular cells, but linear velocity cannot be detected by these cells. So two things that can be detected are static equilibrium, and second thing is the linear acceleration, but not linear velocity. Now, after discussing the utricle and saccule, we will come to the semicircular ducts, and the organ for the detection in the semicircular ducts is known as the crista. And crista, they are associated with kinetic equilibrium, or the equilibrium associated with position. So whenever a person is associated with angular acceleration, the person is bending at an angle, the person is rotating, moving, but the head is having an angular motion, what will happen? The endolymph in the semicircular ducts, what will happen in such a situation is, for example, these are the semicircular ducts. Now they are moving or they are rotating in the direction of motion. But the fluid in them, because of its inertia, it will try to maintain its original position. When it will try to maintain its original position, the outer body is moving, but the inner fluid is trying to maintain the position. So it is like, for example, this is the fluid, and this is how my whole thing is moving. The semicircular duct is moving. Because of this fluid maintaining its position, it is going to strike with the walls of this whole thing. It will go into the macula, it will go into the duct system. So this is what actually takes place. Now they're saying that what happens is three things that we can detect from this type of graph. This graph tells us that when there is like normal position, like the fluid is there, the semicircular ducts are in normal position, they are not moving, the impulses that are transmitted are 100 per second, and there is tonic level of discharge. But now when the semicircular canals begins to move but the fluid is still in there, what will happen? There is rapid motion, and this rapid motion causes increased discharge. With beginning of rotation, there will be increased discharge, and the impulses per second will drastically increase. It will increase to several hundred per second. Now, in the next thing, what will happen is, within a few seconds, the rotation and the number of impulses that are being discharged will become normal, and then the person will stop rotating. So why is this discharge going towards the negative side, or why is it stopping? It is very important to discuss that. So as I told you, what was happening was that the semicircular canals move but the fluid remains static. But after some time, like after 40 seconds, what will happen? That the fluid will also start moving with response to the semicircular ducts. The semicircular ducts are moving and the fluid is moving as well. Both of the things are moving with that. So first few seconds of rotation, back resistance to the flow of the fluid in the semicircular duct and past bend of the copula causes endolymph to begin rotating as rapidly as the semicircular ducts. So we can characterize it firstly with the motion of semicircular ducts only. Secondly, semicircular ducts plus the fluid, both of them, they start rotating in the same method. After that, what will happen? The cupola of the semicircular duct will, due to its elastic recoil, will return to its normal position. Third will be called the elastic recoil thing, that the cupola begins to stop and it goes to its normal position. And now when the rotation suddenly stops, exactly opposite thing takes place. That now what happened, the rotation stopped, so semicircular duct stopped, but the fluid was flowing. So now it will maintain its inertia flow, and now it will strike again, but semicircular ducts are not moving, but the fluid is still moving. This is the first step of stopping of rotation. In the second step of stopping of rotation, the fluid will stop moving as well. Firstly, semicircular ducts stop moving. In the next few seconds, the discharge of the flow of endolymph, it also will start moving, and cupola will gradually return to its resting position, allowing the hair cells to discharge, return to its normal tonic level. Or even if it does not return to its normal tonic level, it is going to return to its region where it gives no impulses at all. And what will happen? In one direction, in the one direction in which the semicircular duct started moving, there was, what was happening in the movement of the semicircular ducts, what was happening? There was a change in position in both the things, and there was increased tonic and increased number of impulses being sent. After that, what happened? This whole thing, this whole paragraph, we can call it the area of the elastic recoil, in which the copula returns to its normal position. And when there is stopping of the rotation, what will happen? The semicircular ducts have now stopped moving, but the endolymph was flowing. Because of that, now the impulses are being sent, but in the opposite direction. After that, what will happen after some time, that now the semicircular ducts plus the endolymph, all of them have stopped moving, as a result of which the transmission in the opposite polarity will take place. But when both of these things will stop moving, there will be no signal sent at all. So impulses will become zero at this point, and hence we will be able to know that this rotation has stopped. So we can write all this mechanism over here. So this is the whole mechanism. Normally 100 impulses per second are going. In the beginning of the rotation, semicircular ducts move, but the fluid does not move and it maintains its inertia. But because the semicircular ducts are moving, so there is some sort of impulses are increasing. After a few seconds, what will happen? Fluid plus the duct, both of them start moving. So there is rotation and there is some type of like signals being generated. So they will reach the peak level at this point. After that, what will happen in the middle? Elastic recoil begins. The elastic recoil is going to cause the depolarization to move towards the hyperpolarization time. After that, the semicircular ducts will stop moving. When the semicircular duct will stop moving, we come to this position like this. After that, but the endolymph keeps on moving because of its inertia. After some time, and during this time, what will happen? Because of this inertia, what will happen, that the motion between the two of them is still taking place. So now the negative polarity impulses will be generated. After some time, the semicircular ducts plus the fluid, both of them stop, and they come to the position of 0 impulses per second. So we can all call on to that. So this was beginning. This was the normal position. This was the beginning. Semicircular ducts, fluid not moving. Semicircular duct and fluid both moving. Elastic recoil. Semicircular ducts stop moving. Semicircular ducts plus the fluid, both of them stop moving. So this is how the whole mechanism takes place in our body. Now we will talk about the predictive function of the semicircular duct system in maintaining the equilibrium. They are saying that the semicircular ducts are associated with the kinetic position, the angular acceleration, but they cannot detect the direction of motion. For example, if the head is rotating like in vertical position, horizontal position, the semicircular ducts cannot do that. The position is detected by the macula, and semicircular ducts, they are going to detect the angular acceleration only. Now, for example, they are saying, they are giving a situation that a person is running, and due to its forward running, during the forward running, the person tends to change its position. For example, a person was running like this, and in this situation, they try to like bends like this or changes its direction of motion. When there will be a change in the direction of motion, it will be detected by these semicircular ducts. The semicircular ducts will detect the whole thing, they will apprise the body, and they will do the anticipatory correction measures. So semicircular duct has one thing which is known as the predictive function in respect to that. But this rapid change of the motion, this rapid change of angular acceleration, it cannot be detected by the utricle and saccule and their macular cells, because they are associated with the static equilibrium. They are associated with whether the person has stopped moving or started moving. They can only detect the change in the motion, but they cannot detect the change of direction of motion or angular acceleration. So that is why the anticipatory adjustments can only be made by the semicircular ducts, but not by the utricle and saccule. Now they're saying that the cerebellum, it is closely associated with the detection of the type of signals that are being sent by the semicircular ducts. And if we remove the flocculonodular lobe of the cerebellum, what will happen? Normal detection of semicircular ducts and its equilibrium mechanism cannot be detected. Normal detection cannot take place. But because the flocculonodular lobes are not associated with the macular signal, they are not associated with the static position. So static position, or we can get apprised that a person has stopped moving or started moving. We can get to know about that. But the direction of motion of that object, the predictive function, the angular acceleration thing, it cannot be done because the flocculonodular lobe are associated very closely with the semicircular ducts and the function of the semicircular ducts, that is to apprise the body of change in the kinetic position or angular acceleration.